This is our DNA and RNA model. First for DNA, it consists of sugar phosphate backbones, which are shown right here. And then our nitrogen bases are guanine, cytosine, adenine, and thymine. And those are the straws. And then our hydrogen bonds, A always goes to T and C always goes to G. And those letters replicate these words. So this is our RNA model, and it is a single strand. And the straws are sugar phosphate. Nitrogen bases are uracil, adenine, guanine, cytosine, and on the DNA model it's thymine, but when it is RNA it's uracil instead of thymine. So our hydrogen bonds are A and U, C and G instead of A and T, it's U.
Okay, so I'm going to be talking about DNA replication. There are 3 steps of DNA replication, and that is unwinding, pairing, and joining. So I'm going to go through and define all of those. So unwinding is the DNA helicase unwinds DNA by breaking the hydrogen bond. Single-stranded binded proteins keep the DNA strands separate during replication. And RNA primase adds a short segment of RNA primer on each DNA strand. So that's unwinding, that's the first step. And then there's pairing. The enzyme DNA polymerase adds appropriate nucleotides to the new DNA strand from the 3 ends. The leading strand is built continuously, the lagging strand is built discontinuously in small segments called Okazaki fragments. The third step is joining. In joining, the DNA polymerase comes to the RNA primer on the DNA, removes the primer, then fills the place with DNA nucleotide once replaced. DNA ligase lines up with the 2 sections. So we have just defined the 3 steps to DNA replication. And okay, so DNA replication takes place in the S phase of interphase, and the purpose of DNA is to produce 2 identical copies of DNA. So what we have here is a labeled picture of what DNA replication looks like. You can see like a lot of things in it. There's the replication fork. It's like where the DNA divides, and that's like right here and stuff. So, and then you can see the DNA polymerase and the RNA primer is like right there. And then there's that word that I can't pronounce and stuff, and then there's the DNA ligase right there. And yeah, so that's basically how it happens. That's like just a labeled picture of it. So the next thing is hydrogen bonds, which as you can see we like labeled over here. A goes with T and C goes with G and stuff. And then there's nucleotides, which is like what makes up DNA. So there's the phosphate group, which would be like right here instead of the phosphate group, and the deoxyribose sugar is like right here and stuff. And then we have the nitrogenous bases, which are the straws. And then DNA is replicated by making a strand that is complementary to each single strand. And yeah, that's basically how DNA replication happens.
So now for our extra credit part, we are going to be doing protein synthesis. And there's 5 steps to protein synthesis, and the first one is unzipping. And DNA double helix unwinds to expose a sequence of nitrogenous base, and hydrogen bonds are broken. And then step 2 is transcription, when an mRNA copy is made from the DNA. So that's AUG, UUG, and AAU are mRNA codons. Then step 3 is translation, and that is when an mRNA codon or an mRNA couples with a ribosome, and tRNA brings free amino acids to ribosomes. Step 4 is elongation, and that is when an anticodon of tRNA recognizes a codon of mRNA. Ribosomes add amino acids to growing chain of amino acids. So they just keep adding on amino acids to a chain and grows. Then the last step is termination, and as it grows, it folds to form a protein and continues to grow until a stop codon is encountered. Then the ribosome will release after that. And then an anticodon is like, so if you had AUG, then A would be U, and then U would be A, and then G would be C, and C would be the anticodon. And then RNA polymerase is an enzyme that synthesizes the RNA strand. And that's it for protein synthesis.